not because the gene is turned on, turned off, or something in the previous stage. I told you a few stages can control the noise by the transcription, by the translation. So to rule out that, we found if we look at RNA label, basically the label are all similar. So it's nothing to do with transcription. But if we look at protein label, actually the protein label is reduced. We know that things happen in the protein label. And we also show the protein stability actually is affected. So it's what we exactly want to see is that's happened after the protein got translated, the folding part could be the problematic part happen inside the cell. So after we demonstrate the protein stability actually is affected, and that's a key protein I already told you, the CLA4 is a key protein to determine the cell shape. So we start to think about how we're going to analyze this phenomenon. So we decide to look at individual cells, because I already showed you, I mean, if you look at populations, some of cells are elongated, some of cells are rounded. So we want to see what happened in those cells. So we use a microscope. Basically, this CLA4 protein is a very tiny amount of protein, only occur in a very small part of cell. It's the butt tip. Here you can see some white part here. Those are the protein. So here again, the part physics contribute a lot because those are the bands of the microscopy. So we can look at those very small amount of protein expression, and we can even quantify the protein amount there. And then, because we can quantify the protein amount in individual cells, that allows us to do this analysis. So basically, this is a histogram of those population of cells which have different amount of the CLA4 protein. And you can see for the wild type cell, basically, no matter you treat the drug or not, basically they have similar CLA4 protein amount. But if you look at the cell which the Hisha protein 90 can be inhibited by the drug, you can immediately find these two populations looks very different. The red one is the one have a normal level of Hisha protein 90, and the blue one is the one which have low Hisha protein 90. And this figure is very interesting because when you look at these two populations, basically you can see they overlap a lot. That's the first thing. Basically, these two populations are not completely separate. So a lot of cells in the low Hisha protein 90 population still have the level of the CLA4 which are similar to a wild type. And if we look at CV value, we can find that's the way we quantify the noise level. You can find basically look at the whole population distribution. The low Hisha protein 90 have much noise level of the CLA4. So this gives us the first idea. Okay, actually in the low Hisha protein 90 population, the CLA4 protein level become very noisy. And we follow those cells because using the microscope we can have a live cell under microscope, and we can just follow how the cell grow. And we confirm that exactly if the cell have the low Hisha protein level, most of them will become elongated. And for those normal cells, actually most of them have higher protein level. So they confirm like when we look those populations, in fact, they suggest those populations will have rounded cell and those populations will become elongated. And again, we want to do some biology experiment to show exactly this CLA4 is the one involved. So if we just simply get rid of this CLA4 protein inside the cell, so you can immediately find we saw this CLA4, all the cells will become elongated. This obviously suggests this level of CLA4 actually play a very important role, correlate with the morphology, because once the CLA4 level is completed out, then all the cells will become elongated. And if we do in a reverse way, we just overexpress the CLA4 to make it a lot of protein inside the cell, even in the low Hisha protein 90 cell. You can see now you have more cells become normal, even though some small proportion of the cells still got elongated. So this result lead to another experiment we try to show is actually there are another protein involved, but I'm not going to get to it in detail. So basically in the end, what we show is in the low Higgs protein cell, basically what happened is there are two proteins got affected, CLA4 and CDC28. And both of them got reduced to the low levels. So when the interplay between these two proteins, they create a lot of noisy phenotype. And to confirm that, again, we do some genetic experiment. If we overexpress both proteins I just mentioned, and you just look at the white bar. So basically, when you overexpress both proteins, even in the low Higgs protein 90 cell, they become normal. They are not noisy anymore. All the cells will have normal phenotype. So here is a model, basically. What happened is, very simple idea here is, okay, we think the role of those 14-14 helper, the Higgs protein 90, actually playing a role is to stabilize the correct folding of those key regulators. So when the Higgs protein 90 level is normal, basically all the cells have very above threshold level of those key regulators. So that's why they minimize the variation, so all the cells will behave similarly. They will show the same phenotype. But however, when this helper amount is reduced, then now a lot of those key regulators become misfolded and they become unstable, they lose ability. So they drop the whole amount of activities to below the threshold. However, because the mean value is very close to the threshold, so some of cells still have above threshold level, some of them drop below the threshold, so they become heterogeneous population. And of course, you will say, okay, so what? You see this kind of noise phenotype, but what does that mean? So since I'm an evolution biologist, so I decided to look a little bit further into different species. So we all know actually all the different organisms come from a common ancestor. And during the evolution, a lot of things change in different species. And the idea is that, okay, if some functions actually are important for those organisms, they should be conserved between different species, even though they have been separated for a long time, they already become very different between each other. So that's why, if you look, human cell and the bacterial cell, those from the very basic biological pathway actually are conserved. We share a lot of similar protein or pathway with the bacteria because those pathway or those proteins are very important for life. So the same idea applies here. We think, okay, if we look different species, actually I use a few species that have been separated from the body is about 100 million years ago. That's a long, long time. And we found if we reduce the Higgs protein 90, which we reduce the chaperone, they can all become very similar. They all show the elongated phenotype. They all become heterogeneous. So this experiment just want to demonstrate actually this kind of noisy phenotype actually could be, they may have some biological meaning. It's just not a very side effect of something else. That's the idea we try to prove here. And of course, when we see this result, the question will be, okay, if from the evolutionary view, that looks like this could be some meaningful phenomena, then how does this work? How can this help the population or help the cell to survive? And I already mentioned in the beginning, okay, those elongated cells actually could help the cell to escape the hostile environment. So how can this happen when the cell encounters stress? So here comes another model. This model I already showed you. Okay, in the normal condition, this protein actually is buffered. So right now they have very high level protein and very high level activity. So when the cell encounters stress, what will happen? So the biologists already know when the cell encounters stress, actually a lot of protein inside the cell will become denatured. And when those denatured protein happen, they could be very harmful to the cell. So what happens is those chaperones have to redirect to work on them first, because those denatured protein is going to damage the cell and damage a lot of cellular function. So the cell have to deal with those denatured protein first. So this is what we call titration model, because once the chaperone are redirected to work on those denatured protein, then the amount of chaperone can work on their original client become titrated. So that means now inside the cell they may not have enough T-cell protein 90 to work on those Clay4 or those CDC28. And because those effects we already show when they don't have enough amount of T-cell protein to work on those T regulators, what will happen is the protein level will drop. So the cell will become heterogeneous. This part we already showed in the previous. So now the second part we have to show is whether the stress really can cause the whole thing happen in the way we just proposed. So here just we showed it. So basically you apply a few stress conditions here. I just show you one result here. It's high temperature. So when we treat the cell with high temperature, basically again the cell is one type of stress which causes protein denature. And exactly we found the cell will become elongated and the population become heterogeneous. So it seems to support this model. But however, in order to prove what exactly is like what we predict, we have to show two things. Because if this model is correct, then you predict two things. First is because now the Hitcher protein is titrated to work on those denatured protein, so the protein level of this key regulator should be reduced, just like we treat the cell with a drug in the previous slide. So one prediction is, okay, these two protein, the level should be reduced. And the second prediction is, okay, because now the idea is stress titrate out those chaperone, So if we just give a cell a lot amount of Hitcher protein 90, then maybe they can rescue the phenotype. And that's what we did. So basically, sorry, I need to stop. So basically, we look at the high temperature cell. Indeed, the Clay4 and the CD28 protein amount is reduced compared with the cell growing normal temperature, and this is just quantified data. So this support our idea first that exactly happen is those key regulator is down. And secondly, if we overexpress the Hitcher protein 90 cell and then put the cell at high temperature, then the cell become normal again. So we know the titration model is right, because when the cell encounters stress, basically they just don't have enough Hitcher protein to deal with those key regulators. So right now, when we give a lot of abundance Hitcher protein 90, then they can have the ability to deal with them again. So now the cell become all-rounded. This is the white bar shown here. So basically, that's more or less the story we have in the lab. So the idea here is basically we show, okay, other than transcription, other than the protein network, there's one more step which can introduce a lot of noise inside the cell. And this kind of noise because due to the protein folding. And the protein folding is got regulated by the chaperone, which is the helper of the protein folding. But, and other than this kind of noise, we also show basically this kind of noisy population noise actually could be they can respond to the environmental stress, because I already showed you when the cell encounters stress response, they show up the heterogeneous phenotype. But what does that mean here? Because, I mean, is this really beneficial to the population? So we have to go to the idea is what we call a bad hedging strategy. So what is bad hedging? Basically it means when the cell, if the cell is living in a very fluctuating environment, that means the environment condition change a lot. So sometimes the cell cannot respond to the change very quickly. So in that case, maybe the most of the population will just extinct because they encounter those harmful environment and they cannot adapt to them and they can respond quick enough, so all of them will die. So if the cell use a strategy we call it bad hedging, that means bad hedging is just like when you are invest your money, instead of putting all your money in one company, you invest your money in different stock and different company, and then even one company got bankrupt, you are not going to lose all your money. And that's the idea for the microorganism too. So what happened is, okay, right now the cell decide to develop a different state of the fitness, and this kind of fitness are all good for one condition and maybe bad for another condition. So in the case when they encounter those environmental fluctuation, so there's always a proportion of cell will survive under those kind of changes. And the filamentous form actually fit this bad hedging idea very well, because what happened is for those filamentous form is, I already told you, the filamentous form of these cell, they can push the cell away from the local environment because the cell cannot move. So they need some way to escape from the local environment, and the filamentous form can help the cell to do that. So they can go push it away. But on the other hand, when the cell becomes filamentous form, actually they grow much slowly. So actually, if you say the cell division time is the way to compete with other individuals, those filamentous form actually is not so fit, because compared with those round shape, they cannot grow so quickly. And also when they become filamentous, another limit is right now all the cells seem to be more linked with each other, and if they cannot be separated very easily. So if there's some other physical force to stress those conditions, maybe they will die altogether too. So basically, the filamentous form has some advantage in one condition, but disadvantage in the other condition. So they fit the bad hedging idea very good, very well. So basically, in the end, I mean, I'm going to suggest, I mean, this kind of chaperone, or what we call the protein-folding helper, actually could be a very important regulator of those noise inside the cell. And I already told you in the beginning, because I mean, those chaperone actually have a lot of target inside the cell, and a lot of target actually are the key regulator. So the morphological change is maybe just one small aspect of those noisy phenotype inside the cell. Maybe there's a lot of different things can be regulated by the cell. So I think for the first two questions, I already more or less provide some answer for that. So basically, the biologists already found there are several different ways the cell can produce the noise, and those noise sometimes could be very beneficial and could be very useful for the living organism. But I mean, actually, the tougher question for the biologists actually is, I mean, imagine, I mean, if the cell have a lot of regulatory pathway, they can just regulate the things. Why the cell want to use noise to deal with the condition? So that's the first question negative puzzle us a lot, because I mean it seems to be a very primitive way to regulate things. So why the cell want to use noise instead of using those precise regulation? And second is like under what kind of condition actually using noise is better? And this is again a big question actually. Is for us it's difficult to answer, but maybe physicists have more clear idea what can be those advantage to use noise. And another big question is, I mean, even though we know the protein network and we know the transcription, and now even the protein folding can create the noise, but whether there's other sources of noise or not, it's still worth of thinking. And how the cell integrate different level of noise, and to use those kind of noise on a good way, not just the noisy things the cell try to get rid of. That's a big issue for biologists because we can think about, okay, cell, if imagine you are designing a cell, maybe you want to just perfectly design everything so there's no noise at all. But since we already found the cell have so many different levels of things they can create noise, how do they integrate different level of noise? And in the end, they still can direct those noise in a good way and without killing the cell. That's a big issue. I think we have to think about it. So in the end, basically I want to acknowledge two graduate students working on this project, and they are just two students working in my lab, and also the funding resource is National Science Council and Academic Income. And I will stop here and take questions.
Thank you for that interesting talk. Questions?
Quick question for photophysics. There could be different forms of noise and different versions of noise as a rule have different even information theory. So in your experiment, did you ever measure well the distribution of the noise to identify, for example, a certain is not Gaussian, it is Poisson, compound Poisson or something like, because physics of all these noises are different from information attribution point of view also.
Well, basically, we didn't have those information, to be honest. And again, since this project, we just got those data very recently, so we just identified the sources from the 1440, but we haven't expanded to look at other level of noise yet. And one of the reasons is because people already show the other level of noise actually can contribute in the other pathway, so that's already well studied. So that's why we didn't pay much attention to that. But again, just like I mentioned, the final challenge, the big challenge will be, this should not be the only source of noise because there are different levels of noise. And how can they integrate different levels to become the final phenotype? That's a tough question for us. And we are always thinking in a very reduction way. It's like, okay, we just figure out one source and we think this can correlate with the phenotype. But I believe that's not true because this source may be the major contribution. Maybe it has major contribution, but other source of noise should be also considered. And how to integrate different source of noise, I don't think anybody can have done that yet in biology. Yeah.
Second question. Actually, what is the life in understanding, modern understanding? What are stem cells? What is the cancer in all this situation? We have both noise and also some risk. So it's very, very interesting to look some correlation of effect of noise with the risk. Have you ever considered in your experiment also some risk or only just the noise?
I'm not sure I really get the question you mean to introduce noise in the other part or consider?
In your cells you are looking some cycles and look also noise related with different rhythms and so on. It's just general observation that in all the biological such interesting situation, noise in some sense interacts with risk. And actually some sense life arose due to this interaction of risk with the noise. So in your experiment, is there any situation to look how noise depends from different rhythms in your cells or not? Are there any oscillation in your cells?
Yes, we do. So we do look so basically in our experiment we try to control the cell in a condition which we can eliminate other source of noise. Is that what you're asking?
Okay, it is more detailed.
Okay, sorry.
Right, more questions? Yes.
That's okay, I have a loud voice.
Okay.
E-SHOP 90, what has that been studied in other cellular systems? Does it?
This is a very common chaperone exists in all the cell, all the entire
So
cell.
is elongation the consequence from alteration of the E-SHOP protein 90? Is that the universal consequence?
Well, in our lab we show is at least for the Easter cell is a universal consequence. But I already mentioned the H-90 can regulate 20% of the other protein inside the cell. So when other people look, they can see a lot of different phenotype when they reduce H-90. And that's why it's a very important protein. Even though the people look at cancer cell, they can find that H-90 is related to cancer cell. Yeah, but for the morphology, I think it's, at least in our lab, we show in Easter cell, the morphology is universal phenomena, and this pathway actually is also conserved. Some of the components actually conserved in other eukaryotic cells. That part is already proved. Yeah, but it has other function for sure. Yes.
More questions? Yes.
Thank you. So thank you for this interesting talk. So somehow I wish to get more confirmative definition of the noise. For example, if I and you looks different,
Yes.
is this due to noise? That's the meaning of noise. I want to confirm this.
Okay, so there are two sources. At least, like you and me, the genetic material is already different.
Different, yeah.
So that's part make the difference between you and me.
Mm-hmm.
But if you look at twins,
Twins,
basically their genetic material are the same.
Mm-hmm.
And then people still can argue, during their grow up, maybe the environmental stimulation is different because they may have different nutrients, they may have different way to grow, so they're still a little bit different, come from environmental. So that's the second source of noise. And the third part actually is the intrinsic noise. That's what we are talking about here, because basically every cell in our experiment have the same genetic material, and then we treat them exactly, and we try to control the conditions so they are all exposed to the same environment. Mm-hmm. So there's no, we try to minimize the external noise. So basically now we are talking about the intrinsic noise come from inside the cell. So while the same materials inside every individual cell, they will make them look different. That's the noise we are talking about in this talk.
So with this viewpoint, will you say cancer cell comes from noise? Because cancer cell comes from the same genetic set in my body, right?
Well, I mean, for a long time, in fact, biology have the opposite view. For a long time, people believe cancer cell actually come from genetic mutation. They think cancer cell have some mutation occur, and then they produce a cancer cell which is different from other cells inside the body. But I already showed you there's one paper published not so long ago. They showed, in fact, even the same cancer cell, they can switch to different type of cancer. So that means there's no mutation. That must be come from something else, and most likely those are intrinsic noise inside the cancer cell. So in that paper, I think they try to demonstrate actually intrinsic noise can make cancer cell behave differently. And actually that's quite new to biologists, because for a long time we just think cancer is just the one carry different mutations, so they become cancer. Mm-hmm. But now they try to show, even though they have no new mutation, the cancer cells still can switch to different type, and then make it more scary. Yeah.
Okay. So my last question, I would like to follow the first question. That I think the main discrepancy between biology and physics is that how to quantify noise. Okay. I think that's the main source of difference, because as he said, in physics we can analyze the distribution of noise and say, well, this is Poisson noise, so like from lasers, from optics, the noise has a distribution.
Yes.
However, how can you quantify noise distribution in such a cell experiment? I mean, quantify noise. I guess that's the main.
So that's the things. I also think it's like the way we quantify is very primitive. Because I mean, the way we only can, the best we can try is to control all the factors we can control, make them grow in the same condition, and using the cell of the same stage. And then the next step is we need a very sensitive, maybe like a microscope, so we can look at the protein amount. or we can even nowadays